Gardnerella Vaginalis. Either it is our friend or enemy. Introduction. The timeline of Gardnerella Vaginalis or GV. In 1953, GV is first described. In 1955, GV gets its name. In 1981, the term bacterial vaginosis is introduced. GV naturally habitats the human vagina. Hence, it is the normal flora of the human vagina. The morphology of GV includes gram-variable, pleomorphic rods, and also non-motile. Apart from GV, Streptococcus species can also be the normal flora of the human vagina. However, these two species can be distinguished. GV and Streptococcus species are also correlated. Simply, when the number of GV increases, the number of Streptococcus species will also increase in cases of infections.
Prevalence.
From several studies conducted, 20 to 40% of women were identified positive with Gardnerella vaginalis infections. Bacterial vaginosis clinical diagnosis can be done by using Eugen scoring system or EMSEL clinical criteria.
Gardnerella vaginalis cytomorphology. Characteristic of Gardnerella vaginalis. First, filmy background of small coccobacilli. Second, a layer of coccobacilli along the margin of the cell membrane. And third is absence of lactobacilli species. Next, the background characteristic. First, the small coccobacilli will form a granular blue background or sandy background on conventional smear. Second, in liquid-based cytology, the background is more cleaner than the conventional smear.
Function of Gardnerella vaginalis. It maintains the pH level depending on its growth and prevents infection through its pH level.
Next, origin and causes. Origin of Gardnerella vaginalis and community state types. The causes of imbalance in the normal vagina condition, such as multiple sexual partners began to engage in sexual intercourse at a young age, vaginal douching, and others.
The pathogenesis of Gardnerella vaginalis infection. An infection is established by three virulence factors. First is the ability of Gardnerella vaginalis to attach well to vaginal epithelial cells by forming clumps that attach to the surface. Next is the production of cytotoxin called vaginolysine by Gardnerella vaginalis that helps in adherence to vaginal epithelial cells. And lastly, the formation of biofilm, which is a cluster of Gardnerella vaginalis that attaches to the surface of the vaginal epithelial cells.
So these are the signs and symptoms which can be in general and also based on EMSEL's criteria.
Next is bacterial vaginosis and the complications. Correlation between bacterial vaginosis and Gardnerella vaginalis. The 6 complications of Gardnerella vaginalis.
For lab diagnosis of Gardnerella vaginalis, it is based on microbiological tests except for pH indicator. Another method to detect the presence of Gardnerella vaginalis is by culture method, as Gardnerella vaginalis is a highly fastidious organism. Next is biochemical test, where Gardnerella vaginalis shows negative results in catalase, indole, oxidase, and urease tests. Meanwhile, in starch and maltose fermentation, it shows positive result. Another method to detect the presence of Gardnerella vaginalis is by DNA probe test. It is also known as PD-FMVP3.
For the treatments, there are oral antibiotics which needs to be taken orally and intravaginal antibiotics, which is a gel or cream that need to be inserted into your vagina.
As for the prevention, avoiding any activity that can disturb the normal flora of the vagina can be taken as a preventive measure, such as avoiding vaginal douching.
So the conclusion is Gardnerella vaginalis can be a friend and also an enemy.
That is all from us. Thank you for watching.